Today, we will be exploring how impaired beta-oxidation increases vulnerability to influenza A infection. Firstly, what is influenza, or more specifically, influenza A? Well, influenza, or the flu, is a respiratory illness that is caused by influenza viruses that infect the nose, throat, and lungs. The flu is typically spread, the most, by tiny droplets made when infected persons cough, sneeze, or even talk. According to a study published by the CDC, around 3 to 11% of the US population gets sick from the flu each year. Each of those persons is the most infectious, 3 to 4 DPI, or days post-infection. One of the most common types of flu virus that causes the flu is influenza A. Influenza A is divided into subtypes based on the proteins that are on the surface. The first one is hemagglutinin, or H, and neuraminidase, or N. Does H1N1 sound familiar? Well, H1N1 is a subtype of influenza A. If you're interested, H3N2 is also a subtype. Shown here is the RNP, or the ribonucleoprotein complex, which is essential for transcription, translation, and replication of the flu's viral genome. This here is the M2 protein ion channel. M2 is matrix 2 if you were wondering. This ion channel is activated to acidify the inside of the virus and provoke viral uncoating so that the virus can replicate. Given this, influenza A has a high morbidity and mortality rate that are caused by metabolic disorders, age, diabetes, obesity, and metabolic syndrome. The impacts of influenza A are just one of the things that Sebastian van Liempd et al. Explored, except one small thing is different. What exactly is this research paper doing? Well, Liempd et al. Related metabolic changes with influenza A virus susceptibility using inbred mouse models. They did this by repeatedly collecting and measuring plasma up to 30 DPI. They then ran the plasma through high-resolution liquid chromatography coupled with mass spectrometry to detect and quantify plasma metabolites. Now, if you remember from our last lecture together, metabolites are chemical end products that make up the metabolome. This is a picture from class that I used last time in our last lecture, showing the metabolome and the metabolites. Earlier, I said that inbred mouse models had been used in this study. There were two different strains of mouse models, the DBA2J mice models, or D2 for short, and the C57BL6J models, or B6 for short. These two strains are two of the most commonly used inbred mouse strains in neuroscience research. Previous studies showed that the D2 mice had higher insulin and glucose levels comparatively, which make them predisposed to develop diet-induced type 2 diabetes. In addition, B6 mice readily recover from infection with a low virulent influenza A virus strain where it would be lethal to D2 strain mice. What did they find? Well, the biggest metabolic difference they found was between healthy and infected strains in the levels of malonyl carnitine. Malonyl carnitine is a metabolite that accumulates with disruption in fatty acid oxidation caused by the long-chain acylcarnitine esters having trouble getting into the mitochondria, and by the failure of the ETC, or electron transport chain. Now, if you're like me, I don't quite remember where malonyl carnitine is in these pathways. Thankfully, the research article provided a diagram. Now, we'll circle back around to these for the mechanisms, but down here you can see malonyl carnitine, MCAR, You can also see malonyl-CoA, citrate, and pyruvate, which you should recognize. So a lot of really fun things going on here. This is figure 3 from the research paper. It's a heat map of the percent change from the baseline for metabolites found in the plasma. Now, a red square represents a positive effect, meaning the rats showed improvement, and important metabolites increased from the baseline after being infected with influenza A. Now, a blue square represents a negative effect, meaning the rats declined and important metabolites decreased from the baseline after being infected with influenza A. However, what does any of this mean, and how does it tie into beta-oxidation? First, let's do a brief overview of beta-oxidation. Here we have our handy-dandy metabolic map. You've seen this map many times in this class before. But before we move on, your first quiz question is to tell me where on this map beta-oxidation is. You can tell me a vague area, or if you like, you can highlight it and submit the picture with your answer. In addition to labeling where beta-oxidation is, tell me how some of the previous pathways we have learned tie into beta-oxidation. We recently started this area in class, so if you need help, the Chapter 21 PowerPoint might be a great place for you to look. Earlier, I mentioned that the study revealed that levels of malonyl carnitine were affected by infection with influenza A. In D2 strain mice, the level of malonyl carnitine increased 50 times the amount present in the baseline, which, for this part of figure 6, is a healthy B6 strain mouse. This is what beta-oxidation and fatty acid oxidation should look like for a healthy B6 strain mouse. This mouse takes in food, and the glucose gets turned into pyruvate during glycolysis. The oxaloacetate supplied comes from pyruvate by the enzyme pyruvate carboxylase. Following through the TCA cycle, citrate is made and moved out of the mitochondria. Once in the cytosol, citrate is broken down by ATP citrate lyase into acetyl-CoA and oxaloacetate. That oxaloacetate can be converted to malate by malate dehydrogenase, which can re-enter the mitochondria to be used again. Acetyl-CoA is then transformed into malonyl-CoA by acetyl-CoA carboxylase. From here, malonyl carnitine is produced here, and malonyl-CoA can move its path to carnitine palmitoyl transferase 1. This is an enzyme in the outer mitochondrial membrane that converts long-chain acyl-CoA species to their corresponding long-chain acyl carnitines for transport into the mitochondria. This is where problems arise when the mice are infected with the influenza A virus. This is the beta-oxidation pathway for a mouse infected with influenza A virus. As you might remember from previous slides, malonyl carnitine is a metabolite that accumulates when long branched chain amino acids cannot enter the mitochondria. This decreases mitochondrial beta-oxidation. This decrease is offset by increased TCA cycle and peroxisomal beta-oxidation, since energy is in higher demand when you are sick. Now, malonyl carnitine is a proxy for the production of malonyl-CoA. Malonyl-CoA is a strong inhibitor of carnitine palmitoyl transferase 1, CPT1 here. Malonyl-CoA binds to CPT1, which initiates the uptake of fatty acids by the mitochondria. If there's a decrease in malonyl-CoA, CPT1 can't be inhibited. This blocks long-chain amino acids and fatty acids from entering the mitochondria. This obviously decreases beta-oxidation in the mitochondria, but it also increases the export of oxidized fatty acids and increases acylcarnitine levels. This can have devastating impacts on not just mice, but humans as well. Now, before we move on to how decreases in mitochondrial beta-oxidation, increases in the export of oxidized fatty acids, and increases of acylcarnitine levels impact both humans and mice alike, I have your second quiz question. For this question, I want you to look into how malonyl-CoA inhibits carnitine palmitoyl transferase 1, or CPT1. This one is CPT1, and this one is malonyl-CoA. I want you to give me specifics on the type of inhibition this causes and what changes are made when CPT1 is inhibited. These changes can include and are not limited to physical changes, chemical changes, and rerouting changes in the pathway for beta oxidation. I know we haven't talked about specific inhibition types in a while, so the class PowerPoints from Chapter 6 and 13 may be of use to you. This book may also help you answer the question. Now, the decrease in beta oxidation can have devastating effects on the body. A decrease in mitochondrial beta oxidation backs up the use of oxidized fatty acids, causing an influx of them to be sent out into the cell. Increased fatty acid oxidation has long been considered a culprit in the development of obesity and diabetes mellitus-induced cardiomyopathy. Diabetes mellitus-induced cardiomyopathy is a specific form of heart disease that is caused by abnormally high blood sugar. The decrease in mitochondrial beta oxidation also increases the levels of acylcarnitines. A variety of age-related diseases have been tied to an imbalance of acylcarnitine levels. Some of these diseases are cardiovascular disease, type 2 diabetes mellitus, osteoarthritis, chronic obstructive pulmonary disease, macular degeneration, glaucoma, and Alzheimer's disease. In addition, abnormal acylcarnitine levels are associated with the activation of inflammation and mitochondrial dysfunction. Those contribute directly to the process of aging. Also, since the mitochondria and peroxisomes are involved in the antiviral innate immune response, the increased metabolic stress on these organelles by influenza A leads to high uncontrolled virus replication, specifically in the study's D2 mice. In summary, impaired beta oxidation increases your likelihood for disease, lower immune system regulation, and unfortunately, in a lot of cases, like for the D2 mice in the study that were infected with influenza A, death. These were just a few of the references I used. And again, here are the full quiz questions if you end up needing them. Thanks.